again, thanks for speaking to us. And the screen is yours. I don't know if you can share already. I think you can.
I'm trying to share, but it's complaining here that I need to change my preferences, and as usual, we have.
Okay.
Why is that so? I always do it in Zoom and it's all right. So is it because of Gather?
No, it shouldn't be. You should be able just to press share screen and then.
But I have had problems with this before, and I sometimes have to leave one of the platforms. So if you're now on Zoom, Siddhartha, I suggest you leave Gather, and then you
close the window, close the Gather window just in case.
You just can just close it. That should be sufficient.
Yeah. Let's try again. Or maybe I need to access again. No, not at all.
I don't think so.
What if I email you my PowerPoint presentation?
Absolutely. If you share with me,
otherwise we're gonna spend time. Yeah. We're already very late. I'll send it to your Neuro account.
Okay.
So you can't share your screen, is that it?
Yeah, for some reason.
Siddhartha, meanwhile, try to get out and enter again. It just goes straight into the Zoom. Zoom. I'll send you again the invite, and then.
Okay.
Okay. And then you can see. Right. Sent. Did you send me your presentation?
Yeah, I'm sending it to you. It's only 114 megabytes.
I don't know if it works, but perhaps, I don't know if Draul is ready. Do you want to switch?
Well, I have to teach at 7. I want to see. I want to see Draulio anyway. So I just sent you the file, okay?
Okay.
And I'm gonna do what you just said. Get out and get in. Okay. See you in the next life.
All right. So meanwhile, Ernesto, you entertain the crowd, and I will try to get Siddhartha's presentation up.
Sure, it will be my pleasure. So I have here with me João from Portugal, that I can also summon to, and Eduardo there. I can see Rafael também there. Marcus he tells me he's in Stockholm. I haven't seen him in a long time. I would love to hear him, to see him on camera. Marquinhos, turn on your camera, man.
It is on.
Move yourself. Ah, okay. I forgot you're Austrian. I forgot you have to maintain a snobbish, sphinx-like stance.
What does that mean?
You still? I don't know. I'll leave it to your interpretation. But hey, are you still in industry? You were telling me.
Yes.
And how's it going?
Things are good. Yes.
Nice. Yes. Sphinx-like, sphinx attitudes. I think you're doing it very well. But
we're very close to science, so I still do science.
But where are you working at, if I can ask?
Yeah, I cannot reveal the company now, right?
Wow.
No, it's a big biotech company in the single-cell RNA sequencing and spatial sequencing field.
No, now we need the CIA.
Good to see you.
Or even worse, Elon.
George.
I'm not in sales, guys. I'm in business. But I'm very happy to see you all, and I got invited here through Kia, and I'm very happy that I could join here and see what everyone is doing, where I have my roots, right? As a
hey, Max, you know, if you put this background there, you look bolder, man. Because it's actually,
I am bold.
Yeah, you are, but you know, if you actually take the background, put a white background, maybe it kind of it gets more.
Switch on your camera,
Look like you
Richard.
have no neck, Marcus. You lost your neck.
No, I mean the
Oh, now
bed,
you
man.
have
I'm
a
just
neck.
like
Now you have.
finally in the in the comfort of my house.
What happened? Is
I
it
mean,
your
I
neck
have the age
show?
of being Mark's father.
So I'm trying to upload here. I'm waiting for Siddhartha. Oh, there you go.
Is it the Ernesto late night talk show?
Yeah, man, this is my future.
Tonight we have Marcus Hirschel.
You know, just before Siddhartha starts, I remember one thing is like when Marx was here, he used to get like he used to buy old bikes and sell, you know, 4 times the price to people in ICE. And then it's like, you know, when Marx is interviewed in Forbes, they say, "Oh, do you remember how you did your first $1 million?" And no, the first $1 million I don't remember. I remember the first guy I rip off with my business attitude.
Hey, I just have, before my show, the first of my show ends, I have to say this, that I'm sure everyone has had this experience. When we want to show how hardcore ICE was, we said that we even have someone doing experiments in the bathroom and publishing in the best journals. So Richardson, thank you. You are the poster boy for ICE, at least in my book.
I'm touched.
The toilet boy.
I'm touched. I prefer toilet because Paulo Vianney, my ex-postdoc, actually used that as a toilet.
Hey, no revelations on YouTube.
You're running late.
Are you
Let's
back?
You're
Yes,
right.
exactly. Please, let's come back to it. Siddhartha, thank you so much. I see you managed to upload your screen. Please feel free. Thank you again. The floor is yours.
Guys. It's a pleasure. I'm going to be a little fast because I want to see everything that Braulio will tell us as well. And this work is about LSD, but I'm going to talk about other things and related things because of the psychedelic field. So classic psychedelics are those structurally related to serotonin. And LSD is one of those. Psilocin, psilocybin, DMT, 5-MeO DMT that Richardson talked about, and DOM, DOI, which are used a lot in the pharmaceutical companies and all part of this. They discovered it's not correct because it was. You can't hear me. Really? You can't hear me?
It's oscillating your connection, Siddhartha. Now you're mute.
Can you hear me?
Yeah.
Yeah?
Yeah, now it's better. Continue.
Okay, so I was saying that LSD was not really discovered, but because it was invented, it was created from chemical synthesis. In 1938, Albert Hofmann, working at Sandoz, big pharmaceutical company, was synthesizing several variations, using different groups to put to make amides. And then he discovered this particular one, the 25th, that had the diethyl antenna attached to the molecule. And then it didn't seem to do anything. It was just shelved. And the procedure then was once shelved, forever shelved. But then for some reason, he felt he needed to resynthesize it. And that was totally against the company's procedures. So he really went against the rules, resynthesized in 1943. And then he accidentally got contaminated and then experimented very strong effects. And then a few days later, he asked for help from an assistant, and then decidedly any kind of effect. and that was 250 micrograms, the famous Hoffman heroic dose. And then he took it and then went back home using a bicycle and had this very famous acid trip. And these are his words: little by little, I could begin to enjoy an unprecedented colors and plays that persisted behind my closed eyes, kaleidoscopic, fantastic images surrounded me, alternating, variegated, opening, closing themselves in circles, exploding colored fountains, rearranging, hybridizing themselves. So this is a chemist, a very serious guy that discovered these amazing effects at very, very, very low dose, unconceivable. This is a series of sketches performed by, you can't really see what is written below there, right? Can you see who did it? Can you see the name? There you can see here now. Okay. So that was done by Oscar Gianni. He was a precursor of LSD therapy. He did that 10 years before Tim Leary, and then he stopped once it became illegal. And this is what happened in the 60s, right? People started to take a lot of LSD in the United States. There was the hippie movement, the merry pranksters that went through the country spreading acid use. Ken Kesey, an important writer, and Tim Leary in '66, this famous phrase, turn on, tune in, drop out. The Summer of Love, Jefferson Airplane that Richard mentioned. So all that happened in the mid 60s. And then soon LSD was banned. And it began to be banned in California, Nevada, and then it was banned all over the world. Many years later, Hoffman would publish this book called LSD, My Problem Child, because LSD seemed to be a problem for the world. That at least was the discourse. If you see what happened to publications, if you just put the word LSD in PubMed, you'll see that between 1950 and 1992, you just have a big mountain that decreased and it was pretty much inhibited. However, if you look at the whole the distribution of data until today, you see that it's a bimodal distribution. So now it's very fashionable to study psychedelics. This is the cover of Cell a few years ago with crystallography work trying to understand why LSD binds to the receptor and stays, and to the serotonergic receptors and stays bound for so long. And it has to do with this diethylamide antenna that was so obsessing for Hoffman. So why is it that there's this revival? And Richardson already alluded to this. The revival is because these molecules have tremendous importance for medicine. This was already, people felt it in the 50s and 60s. A lot of science was done then, not all well controlled. But then people had to drop this research. And if you look at LSD and depression, you see it's also a bimodal distribution with a very sharp peak in the past 5 years. So it's really a hot topic in science. However, psychedelics were not discovered by Westerners doing science. Psychedelics were discovered by shamanic traditions, by several originary populations, and in particular Amerindians, right? There's a long history of psychedelics as sacred medicine in a variety of cultures. And I show here the great shaman Davi Kopenawa from the Yanomami population, and they use the snuff yakuana that contains DMT and perhaps 5-MeO-DMT to reach the world of the spirits, the so-called Shapiri. And different traditions in the Amazon use a combination of plants, the so-called ayahuasca tea, mixing Banisteriopsis caapi and Psychotria viridis to produce several effects of therapeutic and curative properties. And in Mexico, you'll see similar practices using the magic mushrooms, and I think here of Maria Sabina as an important shaman. So there's a lot of knowledge out of science that was actually that helped scientists in the 20th century to move along, and this is also happening in the 21st century. So could psychedelics be the holy grail to understand and treat consciousness? What is the promise for this kind of research? So the promise is that from plants, from fungus, from synthesis, from animals, also in the case of Bufo alvarius, molecules can be extracted and then understood in terms of how they bind to receptors in the brain and change the activity of neurons, a lot like along the lines of what Richard has shown us. Why is this important? Well, it's important because there's a lot of suffering in the world. This is a map of deaths from suicide. There's a lot of suicide in several countries. Globally, 800,000 people die from suicide every year. It's one of the leading causes of death in young people, and very high levels for older individuals. And depression is also widespread, even though people take a lot of antidepressants, but many people are depressed. If you look at the United States, it's dramatic, the situation. The share of population with depression ranges from between 2% and 6% in the world, and older people also get more depressed. And this is a problem. This is exactly the study that Richard mentioned in his talk. Antidepressants don't seem to, at least the traditional ones, the ones that are used since the 80s until today, don't seem to be really very good at being antidepressant. They are all better than placebo, but with a very small effect size, and with many unintended adverse consequences. Most of the studies are only run up to 8 weeks, and most people taking antidepressants in the Western world are doing it for years, also as combination of medicines that were never really tested. So it's a mess what's going on in this industry, and in fact they are now very reluctant to release new drugs because they have a liability problem. And this problem is so important that actually people in the establishment have noticed, because it's hard to notice things when you are at the center of the hurricane, right? But this is a conversation that Thomas Ensel, who headed the NIMH for 14 years, he had at the Psychedelic Science meeting in 2017. I was there, Braulio was there, and many colleagues, many friends. And we saw when we could listen his own words, and you can go on the internet and you can find this available in YouTube, in which he says, well, psychiatry has a problem because we think we understand the mechanisms and we're helping the patients better. Nevertheless, suicide and depression are rampant. And we need to understand, we need different compounds, and also need to understand what the setting is, the importance of the context of use, not just the substance, but the environment and in the general context. And now, again, it's a good dialogue that we're having here with Richardson's talk. Prozac, or the traditional antidepressants, fluoxetine, the inhibitors of the reuptake of serotonin, they have been used as a drug that is a panacea for depression, irrespective of the context, of the social context, of the environment of people taking it. So in this study here, it was done in mice. It was shown that animals that are in a nice environment, they will show something that looks like an antidepressant effect for fluoxetine. However, if they are not living in a nice environment, the effects can be the opposite. So maybe these drugs are pro-depressant, depending on if the context is negative, is adverse. And that it's a big criticism on this branch of psychiatry, or this flavor of psychiatry, in which there was very little space for listening and talking to the patient, and just on the substance. And now it's becoming more clear that a little substance and a lot of psychotherapy is much better than the other way around. Now, this psychedelic renaissance is not only focused in a few substances, it's really spreading to different substances. Psilocybin is a big, important player, and MDMA is another one. This is a study that is very important, a recent one from the Mithofer couple, and MDMA is now on phase 3 in clinical trial, phase 3 in the United States. It's becoming a major medicine. The Pentagon is buying for sure as soon as it becomes available. They've been interested in talking to the people that are promoting MDMA to buy it for the Pentagon, because they have so many veterans. This is the most important drug nowadays to treat trauma, to treat post-traumatic stress disorder. And a big player in this is ayahuasca. Some people in the psychedelic field say that ayahuasca among the psychedelics is the King Kong of psychedelics. And there were several studies at USP Ribeirão Preto that were preparatory for this one here. And then Braulio and Fernanda Palhano and all this team of people were able to publish the first well-controlled, randomized, placebo-controlled trial for psychedelics for depression. And they had amazing results that were not just fast, but that last for at least a week and perhaps longer. And these results were so that the size of the effect was so big that it could beat the placebo effect, which was very big in this case because of the very nice setting that was provided for the patients. And this study really hit the mark. It was all over the world. It was on CNN, displayed in the main page of CNN. I saw news about this paper from Greece, from India. Very interesting to see something that comes from the Amerindian tradition through science, be important for the whole world. And of course, it's more important here because the setting is so interesting. There's a lot of cultural baggage here that plays, in my opinion, in favor of ayahuasca. People are really interested now about ayahuasca. One interesting thing that I won't have time to touch here is that the long-term therapeutic effects seem to be proportional to the acute perceptual changes during the psychedelic experience. Recently, some people have argued that bypassing the serotonergic receptor 2A, the effect is not there, but some of the cellular effects may be there. And this is also in dialogue with what Richardson said. But however, in several studies, the more the person trips, the better the outcome in the end. And this is interesting because it really vindicates, if it really holds, it vindicates, let's say, the point of view of shamanism, which is that people need to really go through the experience. And then it questions a little bit, it challenges a little bit the approach of anesthetizing subjects so that they don't really experience the trip. Or in the case of the Olson lab in California, the search for compounds that are producing the cellular effects without psychedelic effects is important for healing. And in that respect, Braulio and myself and a team of collaborators, we published this paper back in 2011, and it was the first one that we did together on ayahuasca, in which we searched for some of the neural bases of the images, the sound images that occur during, that some people have when they take ayahuasca. And we found, I'm not going to go into details because you guys know this work, I just want to put it in context. We found that when people were intentionally imagining things, the activation was huge when they had ayahuasca, even with the eyes closed, and a big network of areas involving the visual system, temporal regions related to memory, and the frontopolar cortex related to the task, the intentional task, and perhaps other aspects of the task were activated. So we've been thinking and organizing research around these questions here. What are the mechanisms underlying long-term behavioral changes induced by psychedelics? And can these mechanisms be activated to enhance cognition? I want to pay homage here to Brazilian neuroscience, and in particular to Richardson. These are most of the papers showing that, in fact, psychedelics induce neurogenesis or synaptogenesis or induce plasticity in some way measured by some marker. And now there are two more papers to add here. But if you notice, until recently, Brazil had 4 of the publications, and the next one I'm going to show in the following slides. And Richardson, running on a parallel track, because Rafael's work was totally separate from everything we did. He was very generous and helped us do our research as well. And I want to say it publicly. Viu, Richardson? And because of this confluence of interests, at some point, I teamed up with Braulio, with Stevens, with Toffoli, and with Amanda Fielding, a pioneer of psychedelic research that is behind most of the research published to this date from the Beckley Foundation. And we decided to pursue a combined effort towards understanding better what LSD is doing to the brain and to behavior. This collaboration involves many people. It involves César Renaud at EMD. It involves Daniel Martins de Souza at Unicamp for the proteomics. It involves Encarni Marcos at Alicante, at the Alicante Neuroscience Institute, if Cristina is still there. So this is a big collaboration, an international collaboration. And we placed a preprint in December 2019. Now we have more data, and we have a new version of the paper to be submitted soon. Some of the work was the work of Felipe Cini during his master's, and he was studying rats. The first thing he did was to inject rats with LSD or saline, wait an interval of 3 or 9 days, and then habituate them to an arena, and then expose them to the object recognition task. Basically, the animals were exposed to two identical objects, and 30 minutes later, they received a different object, remeasured the time they spent with the novel object versus the familiar object. What we found is that animals that were treated with saline in yellow, they showed an effect that is known, that the preference for novelty decreases from young to adult and old animals. And we found that LSD increased the time exploring novelty for all animals, but it was not, I mean, if you look at the medians, they seem to all increase, but the effect was not significant for the old animals. It didn't reach significance. It was significant for adult and young animals. We reasoned that perhaps the old animals needed a kick, and could be enhanced. But before that, we searched a little bit the parameter space. What were the doses? We found that the dose with maximum effect for time exploring novelty was 0.13 milligrams per kilogram. So we went for this dose, and we applied this protocol here for old animals, and some of them received either saline or LSD once. Some of them received either saline or LSD 3 times, 3 days in a row, and then they were exposed to an enriched environment for 3 hours a day for 6 days, and finally they were exposed to the object. So the idea here was to boost plasticity using LSD, and then boost the requirement for plasticity by exposing to the enriched environment. And what we found is that old animals treated with saline, they did not cross the dashed line that is a level like a young animal level of exploration of novelty, even if they were exposed to novelty. But in the case of the animals that took LSD and received novelty, this threshold was crossed. In other words, the combination of LSD and enriched environment was able to cognitively rescue these animals. A different part of the experiment that was done at the Ren lab and at Martins de Souza lab in Rio de Janeiro and in Campinas was a combination of brain organoids made from human neurons treated with LSD, and then the proteomics of those organoids. So using LCMS and to do the proteomics, the group identified 3,400 proteins. From this total, 234 proteins, about 6.8%, had an expression significantly modified by LSD. And the enrichment analysis pointed to several processes that were potentially affected, such as DNA replication, exon guidance, synaptic vesicle cycle, the mTOR pathway, LTD, and dopamine neurotransmitter release cycle. How am I doing time here? So the mTOR pathway, which was one of the top 10 processes regulated by LSD in the brain, was significantly enriched. And the percentage of proteins differentially expressed in the LSD treated samples annotated to mTOR was considerably higher than the whole, when compared to the whole human genome. So it seems like this pathway is really enriched in our data sets. So from 0.79% in the background to 0.15 in the LSD treated. This project also involved humans. This is work done by Isabel Wisner as her PhD work under the guidance of Luis Fernando Toffoli at Unicamp, and other people involved in the analysis of the data and the generation of the data, including Fernanda Pagliano. And basically here, what we tested was whether LSD treatment could change the way that a memory is remembered when applied before the training or after the training. Here you see the first task was learning card locations, so pairs of cards, just like the memory game. At 8:30 a.m., at 9:30 they would receive LSD. At 8:30 a.m. they had to recall the card locations, so that was the first assessment of performance. And then on the next day, and then after that they had to copy complex figures and have an immediate recall measurement, and then a delayed recall at half an hour later. And what Isabel found is that there was a significant increase for recalled card locations for the copying the figures, the complex figures, on immediate recall, but not on delayed recall. It didn't reach significance, although there seems to be a trend there as well. I don't have time to talk about the computational work that was performed by Encarni Marcos and César Renaud, but the conclusions for this first part are that LSD affects proteins associated with plasticity in human brain organoids. LSD increases novelty preference in rodents, in rats, dependent on dose and age, with less effects in old animals. The association of LSD and enriched environment rescues novelty preference in old animals. LSD improves visual memory consolidation and recall in humans, and these results indicate altogether that LSD has major potential as a cognitive enhancer. I also want to take a few minutes to talk about the work of Anise Souza, who finished her PhD with us and is now soon, she just got her visa and is going to start her postdoc in Florida. And we're going to miss her very dearly. And Annie, among many things in the lab, she worked with psychedelics. She was recording from rats using multi-electrode setups to record the hippocampus and the prefrontal cortex. And to make a very long story short, just to, in a couple of minutes more, if I have the time, yes. Basically, what you see on the top left is what we call a state map. This is a technique that was developed many years ago in this paper that I mentioned there. And basically, we filter the local field potentials and we plot some ratios so as to discriminate the different states. So you can discriminate quiet waking from slow wave sleep, from alert exploratory behavior, AE, from REM sleep and intermediate sleep, and so on. And what you see there are the state maps of an animal at baseline, and you can see that the animals they go to slow wave sleep, and then they go to wake, and they go back to slow wave sleep again in this black plot. And when after 15 minutes after LSD, they will show a very different pattern that seems to be occupying the REM sleep and beginning of slow wave sleep areas. And then at 30 minutes, it becomes more relaxed. It goes back to the slow wave sleep cluster in the northeast corner. Now, if you look at the plots below, they are plots of speed. It's the same state maps, but with speed plotted on top of them. So if you look at the LSD 15 minutes, you will see that the animals, when they are after 15 minutes after the LSD intake, and they received both intra cerebral injections as well as systemic injections. But if you look at what happens soon after, 15 minutes later, you'll see that the animals have a pattern of a spectral pattern, let's put it that way, that resembles REM sleep. However, they are moving, as you can see from the speed map. So they are moving around, but their brains, at least from the point of view of the state map, seem to be asleep. Why is this interesting? It's interesting because sleep has been shown in the past few years to tremendously promote synaptogenesis, but also the synaptic selection. It seems like slow wave sleep is important to add synapses to the brain, and REM sleep is important to cut 70–80% of those synapses, to kill them, to prune them, and then enrich the remaining ones. So if animals are, as they go through a psychedelic trip, if they are undergoing something that resembles sleep at the brain level, at the oscillation level, at the electrophysiological level, and they're still awake, so it gives more substance to this notion that perhaps it's some sort of dreaming awake. But there's more research to be done. This paper is not published yet. We're trying to get it out before Annie leaves for her postdoc. And the second part of the conclusions is that the electrophysiological changes induced by LSD mimic the spectral profile of sleep, even though the rats are awake. Given the evidence of sleep-induced synaptogenesis, the results suggest that LSD produces acute electrophysiological changes that promote synaptogenesis, which in the long run modify the synaptic landscape so that it becomes more apt to learn from new stimuli. I want to thank the many collaborators in this work. Most of the people mentioned here are in the same publication that we are ready to submit in the coming days. But also want to thank people that helped this general project over the many years, including Richardson, Catarina, Claudio, Rodrigo, Adriano. A lot of, I think we managed to have an environment at the ICE in which we really help each other and want each other to succeed. And this is important if we want to be having a fun meeting in 20 years, 30 years, 40 years. We need to keep it up. And finally, I want to thank the funders, although maybe we should remove the Brazilian agencies that really don't fund science anymore. This is a total collapse of the system, but we hope this is not going to be the case soon. Soon we're going to have some change in the country. Things will get better, have to get better. We need to believe it. Thank you very much.
Thank you, Siddhartha, so much. Brilliant talk, as always. I will open the mic. According to Ernest, not so much, but excuse me. I'm sorry. I'm sorry, guys, the interference here.
Lucas.
So he wants to know when can we go do something. The mic is open. Anybody has questions for Siddhartha, please feel free to. I'm sorry, I got a little confused here with my young child trying to talk to me. No, nobody. Siddhartha, so.
Vai, Tarcísio.
Vai lá, Dudu.
Siddhartha, what do you think is involved there in the age effect in Felipe's data? Why do you think young don't have such the same effect as that we see in older animals?
I think that if you induce plasticity, but the animal has nothing new to explore and already has a very low drive to explore new things, right? An old animal will tend to explore novelty less than 50% of the time, 40%, even less sometimes. So I think that if you are in that situation, you're basically wasting this boost of plasticity. There's nothing it's non-functional, I think.
I see.
So it's more stimulus dependent.
And it's 6 days after, right?
We chose 6 days.
Do you see more long-lasting effects?
Yeah.
Have you ever tested?
We don't know. We don't know.
We
reasoned that if it's something related to synaptogenesis, 6 days would be fine. If it was neurogenesis, we would need to wait more.
Right.
What happens when you give LSD to octopi?
They stay very quiet. And so do birds.
So do birds. Presumably, it has to be mediated by some sort of gene expression program, right? Do you have any thoughts about what is happening and where is happening mostly?
Say it again. I didn't get it. Sorry.
So assuming this is a long-lasting effect, presumably is mediated by gene expression. You're triggering gene expression programs where you normally do not have. Do you have any idea, or do you have any thoughts of what kind of gene expression programs you're triggering and where?
Well, the mTOR pathway.
mTOR.
The mTOR pathway seems to be, well, it's coming from the organoids, so we need to go back to the rat brain. This is something we want to have oxygen to do, is to proteomics the same way we did it in the organoids, to do it in the rat brain. It's doable. Right. It's feasible.
Right. In the short term, or in like both? So you want to do a longitudinal study in which you have immediately after and also 6 days later? Is that the idea?
We don't know, because these things are sort of expensive, and we need funds for that. So that's one of the reasons why we didn't do it so far. And this paper has been out there since the end of 2019, and only now we will send it. We got reviewed, not full reviewed. We got some feedback from eLife and other journals, but it didn't have the human data. Now it has the human data, the human behavioral data. So we hope that this will allow us to get it out. And once we get it out, we may be able to better fund it.
I think there's a question here from Elcio.
Hi, can I talk? Can you listen to me?
Yeah.
Okay. Hi, Professor Siddhartha. Great talk as always. You mentioned the David Olson's lab in California, where they're trying to find psychedelic analogs, but that have antidepressant effects, but without the psychedelic inducing effects, at least in mice. I had seen you before talking about the entourage effect of marijuana. Do you think that something similar is happening with psychedelics, not a pharmacological entourage effect, but more of a psychological one? You need both the therapeutic session and the psychedelic in order to produce an antidepressant effects, or everything reduces to pharmacology?
No, I think your question is great. I don't think it reduces to pharmacology. I think that to believe that the antidepressant effects are just a cellular effect is to not get depression. Depression is not low serotonin, low neurogenesis. Depression is about your relationships. It's about your place in the world, which is mediated by serotonin, by neurogenesis, and so on. So I actually think that the fact that the enriched environment has this synergy with the drug is suggesting that for therapy, the best thing is to combine drug and setting, the right setting. And maybe by bypassing the trip and only enhancing plasticity, you will not get the results in humans as you would like to, exactly because the person did not have to experience this otherness, which is given by the 5-HT2A receptor. So maybe the other receptors, and in fact, we can be talking here about perhaps dozens of receptors involved, at least 3, but perhaps dozens, because it really, the affinities are it's almost like a uniform distribution of affinities after some point. So all those things are compounding, and I get your analogy with the entourage effect. It's almost like an entourage effect of several receptors, and then on top of that, the effect of the setting. And I think that we should continue exploring this synergy, because I think it is this synergy that describes the healing setting that has been functioning for many societies before science. So in that sense, I find it amusing that people are trying to get rid of one of the most interesting things, because of course they don't want to touch there. They want to treat people like they're machines, not like they're subjects.
Fantastic. I know we probably have a lot more questions for Siddhartha, but since he wants to see Braulio and we're running short on time, I would like to thank you once again, and very nice talk, Siddhartha, and thank you so much. And we'll move on to our next speaker, which is another member of our faculty, Professor Braulio Araújo.
Hi, Tarcísio. I'm just figuring out which avatar am I going to present this talk.
Oh, that's very interesting. It's just a T-shirt.
How about this one?
I think that's great.
So Well, thank you, Tarcísio. Thank you, Diego, and all the Brain Institute for this great meeting. I confess that it was a lot better and creative and everything than I ever thought about. So thank you, Tarcísio, for being, and Diego for being for thinking so much out of the box and providing us with this environment, very interesting environment for this gathering. So thank you to Tarcísio and Diego. I also want to congratulate the ICE for the 10 years. It's been a very interesting experience. I've been here since 2009. So I was one of the first professors to be hired here at the university. So it's been a great pleasure to be here since 2009. And in that sense, I also want to thank Siddhartha and Kirsten, particularly Kirsten, who has been facing a kind of rite of passage in a lot of senses, especially being the head of an institute, which is in the middle of a transition, in the middle of a tsunami. So thank you, Kirsten, for all your work and keep pushing the institute. Also, as Claudio mentioned yesterday, another thing I wanted to talk about before I move on to my talk is that Claudio compared or made a parallel between the institute and his kid, Joaquin, who is about the same age as the institute. And when he said that, I mean, I have a daughter that's about the same age as the institute as well, and that rang the bell. So that made a lot of sense. And that made a lot of sense in a lot of different perspectives. I mean, the first perspective that I thought is that I miss Lara when she was 1 year old, as I missed the institute when it was a child. I missed the institute in 2009 and 2010 and 2011 and 2012 when we were going through a lot of difficulties, it's true, but at the same time, it was a lot of fun. So as being a kid is a lot of fun, being at the Brain Institute as a kid, being a child was a lot of fun. And this is part of the fun we used to have in 2010. If you can, maybe you need to pin my video to see the whole image. So basically what we have here is our graduate course. We have a graduate course in Tópicos Avançados de Neurociência. This was the first time this course was offered. So what you see here in this picture, you see Siddhartha, you see Marcos, you see Adriano. In this other one, you see Fernanda, Sergio, Aron, Fabio. So a lot of the people that are here and who were in our the first students of our graduate program in neuroscience. So to me, it's very important to talk about the neuroscience, the graduate program in neuroscience, which was, in my opinion, our first big achievement. So the first students we got was in 2010. And these classes used to take place at the International Institute in Macaíba, under the trees for the whole day. And I can't think of any other discipline or any other graduate course in which I had so much fun as we used to have when we were having these classes. When we went on to the gas station to have lunch, or the baobab, the beautiful baobab that's out there. So anyway, I just the way that Claudio brought that to us was very interesting. The idea of something that's growing out of a rupture, as Sergio mentioned yesterday. something that was born out of a conflict. And that gave actually us a lot of strength, in my opinion. So coming out of a conflict made us, and us back then was basically a bunch of professors who ventured to a city that no one of us was from, Natal. So a lot of brave people that left a lot of things that they had in, like Sergio, Casting, and a lot of us who had previous positions, and that left those previous positions to join this dream that Siddhartha somehow convinced us all of it. Thank you, Siddhartha, for this. So this amazing group of people, and in the time of the conflict, we were a lot weaker than the other side of the conflict. So the strategy that we took back then, and to me is very clear, was the strategy similar to a bunch of penguins, that we knew we were weaker. But a lot of times, basically what we did was to gather together with our backs against the wind. And every once in a while, someone would leave the group to yell and then come back to the group and look for protection in the group. So that made what we are today, I guess. A lot of what we are today comes from this childhood, this very healthy childhood that we had, with a lot of respect, with a lot of trust, with a lot of friendship. And I think that this is what makes, and at least this is the origin of where the ice came from. So I'm sorry for this, but I had to do this. So
I'm getting emotional.
So the idea here of this talk, I didn't really know what to, I didn't want to present, you know, the same things or the things that you know already. So I decided to try to create a story around the data that we have, basically creating or trying to create a parallel between the ayahuasca experience or the effects of ayahuasca with the pandemic, with a lot of the effects that we have been feeling in the pandemics. It's translating a saying of saying that a good carpenter, that all he sees is a nail. So it's in the same sense that you know, we've been working with ayahuasca for so long, and during this pandemic, it's so clear that of the number of parallels that there are between the pandemic and our work. And this is the reason why I decided to organize this talk this way. So basically going through these 10 years of work that we have done so far, but trying to trace a parallel between that and the pandemic. So the first parallel that arises come from a paper that was published in 2017 by Professor Nicole Galvão Coelho and Professor Bernardetti. And basically in this paper, what they've shown is that there is a very promising model of depression in marmosets. And this model of depression consists of isolating marmosets for 8 weeks. This model was published in 2017, and this actually allows us to foresee what's coming. So social isolation is basically the model that all of us have been in for the past year and a half. And what I'm telling you is that in marmoset, 8 weeks is enough to start developing some kind of depression. And we have been seeing that already when we look at this Nature paper published in February this year. You see that there is already a significant increase in the number of depressive patients in the United States and in the UK. In a year, in the United States, for instance, you have a 30% of increase in depression already in 2020 as compared to 2019. So that, and together with the idea that from the Spanish flu, There's very little data on that, but the data we have comes from a Norwegian study that showed that the following 7 years after the Spanish flu, there is a 7-fold increase every year in the cases of depression for 6 years after the flu. So the first parallel is that we have to get ready for what's coming, for sure. So there is a huge surge in depression and anxiety already coming. But the good news in one sense is that the same model that we used, that Bernardetti and Nicole used to show that social isolation is a potential model for depression, these animals were treated with ayahuasca. So they were first used, the social isolation model was used, and then the animals were treated with ayahuasca, and the results were significantly positive in the sense that there was a positive impact of ayahuasca in the symptoms of depression in models of social isolation, which means that in case it makes sense, of using or of bringing psychedelics to this scenario, and the importance of psychedelics in this scenario parallels with the problem with depression. So the treatments that we have for depression nowadays, if you look for a first pharmacological treatment, you will have around 50% of chance to respond to anything we have available in the market. If you don't respond to the first treatment and you look for the second treatment, those chances reduce to 30%. If the second treatment doesn't work and you look for the third treatment, then the chances drop to 10%. And these are the group of patients that we have been working for the past 10 years, which are patients with treatment-resistant depression, very severe depression, very long time courses of depression, and very low response to anything we have available, to a point that electroconvulsive therapy becomes one of the main perspectives for these patients, or ketamine, which just received a breakthrough therapy authorization from the FDA and from Anvisa. So what I'm showing here in these slides is basically what Siddhartha showed already, suggesting that psychedelics might play a very important role in patients that do not respond to the actual treatments, and also to the upcoming surge in depression and anxiety that we expect. Second, the second relationship, which I found really interesting, is the fact that we, besides monitoring the changes in the symptoms of depression of these patients, we also looked for the basis of these symptoms. So the basis of these effects, of these therapeutic effects, so where the effects come from. So we collected a number of markers from blood, from MRI, from psychological evaluation, from the acute effects, and so on. And one very significant and robust result that we found is a significant correlation between the decrease of the symptoms in depression in these patients with treatment-resistant depression with a decrease in the C-reactive protein, which we all know it's significantly related to COVID. That, together with some of the perspective that Richardson mentioned and Vito mentioned also, it seems as though that these substances might conquer other spheres of potential treatment for humans besides mental conditions. So here we do have a very promising perspective of testing psychedelics as an anti-inflammatory substance. And this is something that's in our perspective of trying to use psychedelics as a treatment as an anti-inflammatory. Ayahuasca, as Siddhartha mentioned, is not the first and is not the only one that's coming. So I just want to mention that these past weeks have been very important for the psychedelic science space. These are two papers published in the last 2 weeks in two very important journals. The first one is a New England Journal of Medicine, that compared a treatment with psilocybin against escitalopram or fluoxetine. And on the right, you see the phase 3 clinical trial that Siddhartha mentioned of MDMA in PTSD, which was published in Nature Medicine the past Monday, 2 weeks ago, almost 2 weeks ago. So it's a fact that these substances are coming, and I'm happy to see that the Institute is prepared, and not only prepared, but the Institute is becoming more and more in sync with this wave of good science that's coming. And I believe that this is first promising, and second, a very interesting field of research. The second parallel I want to have between COVID and ayahuasca is the significant increase of introspection that happens during the acute effects of ayahuasca. So introspection is a construct that basically tries to assess the ability we have to be aware to our own thoughts and emotions. So this is the idea of introspection. Basically, how aware are we individually to our own thoughts and our own emotions? There are a number of techniques that basically try to improve introspection, such as meditation, yoga, and some of these. And ayahuasca is one of them for sure. So it's a very convincing effect of ayahuasca to anyone who have taken ayahuasca. That this substance is significant increases your ability to pay attention to your own thoughts and your own emotions, to a point that you'd say the thoughts came very strongly. So a strong thought is a common word that you see in this field. This is one of the things that we have explored significantly, and something that we have a lot of interest in psychedelics. So trying to relate psychedelics with introspection, or trying to understand some of the effects that comes with introspection. And we have started this work basically by having individuals under the influence of ayahuasca coming into an MRI scanner and looking for some of the networks that seem to be modulated by ayahuasca. And here I'm showing basically two of these networks that seem to be very significantly modulated. And curiously enough, these two networks, as you can see on the right-hand side of the picture, they are coincident with the locations of 5H2A receptors, so the density of 5H2A receptors. And these two networks are the Salience network and the Defomo network. So what we have been finding for these two networks is that they show a very robust modulation, both during the acute effects of ayahuasca, which is what I'm showing on the left. This is a work by Fernanda, and this is a collaboration between us and Lorenzo Paschini, who is from UCSF. And basically what we are showing in these two papers is the first one is during the acute effects of ayahuasca. The second one is during the subacute effects of ayahuasca. So in other words, the right-hand side graphs, they correspond to the comparison of these brain network comparing the baseline against 1 day after the session, so not during the acute effects. So what we see here is that even 1 day after the session, so even 1 day after you went through a psychedelic experience, the modulation in these two networks, and here I'm just presenting one of them, the Salience network, is significant. In the case of the Salience network, what you see in red is the group that took ayahuasca. In blue is the group that took the placebo. So there is a significant difference in an increased activity of the Salience network 1 day after the session, which is the same kind of finding that we had during the acute effects of ayahuasca. In other words, an increased activity of the Salience network, meaning that behaviorally, these individuals are more salient to something. So there's something that's very salient. There's some stimuli that even with your eyes closed, that's very salient. To help construct this story, the second network that we also found a very significant and robust modulation, but that happens in the opposite way, comes from this network that's called the Defomo Network. What you see again in the left is the modulation of the Defomo network during the acute effects of ayahuasca. What you see in the right is the modulation of the Defomo network one day after the session with ayahuasca. What you see here is a decrease in the activity of the Defomo network, a significant decrease in the activity of the Defomo network with respect to the placebo. What does the Defomo network, what is the main construct related to Defomo network? Spontaneous thoughts. So there are a lot of evidence in the literature showing that the Defomo network gets modulated by events related to spontaneous thoughts. So what I'm calling spontaneous thoughts is basically things that we are all aware of, but it's very difficult to describe. And for science, it's a lot easier to describe some of the phenomenology behind these spontaneous thoughts, such as mind wandering. So mind wandering is an event from spontaneous thoughts, and is an event that all of us had gone through during this talk, which is you are looking at me, but basically your head is somewhere else. So although, and this happens all the time, actually, there is a very famous paper published in Nature a couple of years ago in 2010, and the title of the paper is A Wandering Mind is an Unhappy Mind. And in this paper, it brings some very convincing evidence that approximately 50% of our time, of our awake time, we spend mind wandering, we spend daydreaming somewhere. The second event or manifestation of spontaneous thoughts, which is a more pathological manifestation of spontaneous thoughts, is what we call rumination. So rumination is a different, it's a thought with a different characteristic. It's frequently observed in depression. So it's a thought that's basically structured in a way that it's always negative. It's circular. So in the sense that that thought never leaves you, and you're always coming back to the same point. All of us have experienced that when we wake up at 3 o'clock in the morning and there is a spark of a thought that comes in our mind. And this spark can be anything, but a lot of times this spark just amplifies into something that you need to do. And this amplification of that sparks just prevents you from falling asleep again. And then 3 o'clock, 4 o'clock, you're just looking at the watch and saying, well, I got to wake up tomorrow. And then you can't go to sleep. And then eventually you go to sleep. You wake up in the next morning and you say, oh my God, I can't believe that I didn't sleep because of that problem. So that is a very significant example of what I'm calling how strong a thought can be. So how much do you believe in, and so how much pressure, how much energy you put into those thoughts. So one thing that's it's becoming more and more clear is that the Defomo network is directly related to spontaneous thoughts. So to processes related to spontaneous thoughts. So in other words, patients with depression who exhibit higher rumination as compared to healthy individuals, they show a higher activity of the Defomo network, for instance. People that mind wanders more, they show an increased activity of the Defomo network. So the idea here is that both ayahuasca as well as meditation, curiously, they apparently decrease the activity of the Defomo network, which makes sense in the phenomenology of psychedelics, in the sense that it's very common to have a feeling that your mind is calmer one day after the psychedelic experience, not necessarily during the psychedelic experience. And actually during the psychedelic experience, this is what we observed. So this is one scale that we use to measure spontaneous thinking in people under the influence of ayahuasca. It's called the Amsterdam Resting State Questionnaire, and it has a couple of factors. So what I'm showing here are 4 factors that I'm bringing here to make my point, which are 4 factors that showed significant changes during the acute effects of ayahuasca. And these 4 factors are visual thoughts, discontinuity of mind, and verbal thoughts. These 3 factors, they displayed an increase during the acute effects of ayahuasca. What are these 3 things related to? So visual thoughts are questions related to, I thought in images. My thoughts came as images and things like this. Discontinuity of mind is saying that my thoughts were, I had rapidly and switching thoughts. My thoughts were not coherent, things like this. So they were discontinuous. Verbal thoughts, I had silent conversation. I could hear my thoughts. And curiously, in the opposite way, planning decreased during the acute effects of ayahuasca. So basically, you're not spending your time in the future when you are under the influence of ayahuasca. You're not spending your time planning the future when you are under the influence of ayahuasca. Meaning that possibly during the experience with ayahuasca, you are really stick to the present, as is the case of meditation, which would be another parallel between meditation and the experience with ayahuasca. So what is our main hypothesis here is that what happens during the effects of ayahuasca is greatly based on the idea that people gain the ability to see their own thoughts. So this is the whole picture that we have been pursuing, and that we have data, and that we are analyzing data, pursuing this hypothesis. So in other words, you have a decreased activity of the Defomo network, meaning that you have an increased introspection, meaning that you have an increased ability to be aware to your own thoughts. Second, you have an increased activity of the salience network, meaning that something is salient, but you have your eyes closed during the experience. So what is it that's salient when you have your eyes closed during the experience? Your thoughts. So, and what we saw here is that indeed, during the acute effects of ayahuasca, there are a number of areas, including the visual cortex, in which we see an increased activity during the acute effects of ayahuasca. The parallel that we found after we published this paper, we found a lot of parallels between our results and the results that people have been finding more and more in lucid dreaming. For instance, this increased activity of the frontal polar cortex, on the right frontal polar cortex that you see here, it's very typical of the increased activity that you find during lucid dreaming. So the perspective that we have here is basically that the brain would not be inventing another mechanism to become capable of creating very perfectly and visual mental images with the same notion of reality that we have right now. It would have a single mechanism, which would be the same mechanism that we have when we are dreaming, when we're lucid dreaming, or when we are under the influence of ayahuasca. So our hypothesis is that these 3 states, they do share some things. So the brain share the mechanisms through which it produces these 3 experiences. The last parallel that I want to have then between the pandemic and our results with ayahuasca comes a lot from what Lucas presented today when we looked at the increased connection somehow that Lucas found in the patients with schizophrenia. So under the influence of ayahuasca, this result that I'm showing you here on the left, I'm showing you the paper that we published in ayahuasca. On the right, I'm showing the paper published previously with psilocybin. So what you have here in blue for the ayahuasca is the group under the placebo. What you have in red is the group under the influence of ayahuasca, similar to what you have in these two circles. On left, what you have is the placebo, and on the right, what you have is psilocybin. Every point, every dot on the circle here corresponds to a brain area, and every line corresponds to a significant correlation between two brain areas. So what this results here intends to show is that the brain under the influence of ayahuasca or under the influence of other psychedelics becomes more disorganized, but disorganized in the sense that it becomes more free and not rigid. It would be something related to an increased entropy, and that's how we have been interpreting these results. So what we see is an increased disorganization, that we are translating this increased disorganization by an increase in entropy that we find during the acute effects of ayahuasca. This acute entropy can move to, so this disorganization, in other words, would be one of the markers behind all the possibilities that comes when you are under the influence of ayahuasca and the type of thoughts that you can have. So when we are in our ordinary state of consciousness, as we are right now, our thoughts and our action are limited to the habits of our thoughts and to the habits of our behavior. What I'm saying here is that under the influence of ayahuasca, these habits of what you could see, of how you could behave, of the type of thoughts that you could have popping up in your mind, they are more related to the type of thoughts that you have during a dream than what we have during this ordinary state of consciousness. So in other words, there are many other experiences that you can have access during the acute effects of psychedelics. And one very significant effect of psychedelics is the idea, and this is translated with in the meaning of the word ayahuasca, which is the vine of the souls. So that means that these substances, ayahuasca and psychedelics, they have been used for many years to have access to what people have been calling different realities. And what is a way to say, well, you have access? What does that mean? I mean, I have access to different realities. Basically, what you're saying is that your brain, or you were more free to have experiences that usually you don't have in this ordinary state of consciousness. And this can scale up to a very significant experience and very typical experience of the psychedelic, of psychedelics, which is what people call ego dissolution or ego death. So it's the idea that during the acute effects of these substances, you have the experience of dying. As a matter of fact, there is a paper published 2 years ago showing that the phenomenology of psychedelics, they are very similar to the phenomenology of near-death experience. So they applied the near-death experience to people who went under the influence of NN-DMT, and there is a significant increase in these scales, very similar to what is to what happens during a psychedelic experience. And in fact, why is it that I'm calling this a parallel? Just because I can't imagine that we as Brazilians have ever faced death in such a close manner as we have faced during the pandemic. So that you have an idea, well, Brazil, we know that wars are the great killers of humans. So the period of humans where we lost the greatest number of people was during the Second World War. Brazil lost 5,000 people during the Second World War in the scenario of 80 million people. But Brazil got involved in a war, the War of Paraguay. And in that war, we lost 200,000 people. I'm sorry, 20,000 people. Most of them from the army. And we're talking about now 400,050 people. All of us, indirectly or directly, have had to face the idea of death somehow. And this is the most, in my opinion, this is the most important thing that we can bring out of a psychedelic experience. In my opinion, the most important thing we can bring out of a psychedelic experience is what Alan Watts tells in this phrase: if you get the message, hang up the phone. For psychedelic drugs, they are simply instruments like microscopes, telescopes, and telephones. The biologist does not sit with eyes permanently glued to the microscope. He goes away and works on what he has seen. So this translates to me what is the essence of the psychedelic experience, is the idea, or is the very clear notion that after you have that type of experience, is the clear notion that what matters is life, is the clear notion, is the energy that you gain in the experience to come back and make the difference. This is what I believe it's the main parallel that I find between the experience with ayahuasca and the experience with the pandemic. A lot of us will come out of these experiences as transformed as a lot of people comes out of a psychedelic experience. Transformed in the sense that they understand that this is what matters. And in this sense, I mean, we are now, the Institute is now coming out of the second Institute as the first Institute came out of a conflict. This second and beautiful Institute is coming out of a pandemic. So I really hope that we can transform this adulthood coming from the childhood of driving in cars, as Claudio reminded yesterday, driving around the neighborhood as teenagers, trying to figure out what we need to do, coming from that and achieving the adulthood with this beautiful building. So I really hope that first we can take care of the building, starting from the bathrooms, starting with the bathrooms. So we know that public buildings, the first thermostat of a good or a building that's well taken care of starts with the bathrooms. So I really hope that first we can take care of our bathrooms and don't leave our bathrooms in the Institute as we have seen in other places. it's a pity that when we get to the adulthood, we become grouchy, we become suspicious, we become full of issues and problems. And we have that in the Institute already, very clearly. We are in the adulthood. We are becoming a lot more grouchy, very clearly. But this is the time, adulthood, although this comes with adulthood, adulthood is the time when we construct things, when we build, and when we make the difference. So I truly believe that the adulthood of the Institute is when we are going to make the difference, is when in this grouchy and more suspicious and more difficult and with less trust, that we can move ahead. That's how adults move ahead, creating their own conflicts. And I believe that we're going to be able to move ahead and do what we need to do, which is what adults need to do in some sense, is to promote the most positive impact in others people's life. And I believe that we can do that directly from, for instance, our studies with psychedelics and depression, and indirectly through a lot of the research that the Institute is doing. So thank you very much. I'm sorry if I took too long, and I'm here open for questions or for whatever. Thank you.
Can I ask?
I have to say
because I need to teach.
Okay.
No, you made me cry, Braulio, again. No, but I want to ask a specific, serious question before I go. At what time scale is the entropy increasing?
I'm not sure what you're.
Like if you measure correlation at low frequencies.
Oh, okay, yeah. So so we're talking about the hemodynamic response. So we're talking about a couple of seconds, right? It's an fMRI data, yeah.
And if you bend it with bigger and bigger bends, what would happen to this entropy?
I don't know. I mean, because we know that entropy, the definition of entropy is based on the definition of the number of states. So it's greatly defined by the number of states. We haven't done it, so it's difficult to say what would happen.
The reason I say this is because you see talks in which people say, "Oh, the synchronization increases," and then no, it decreases, and though the activity increases or the activity decreases. It depends on what time scale we're talking about, right?
Yeah, well, we're talking about correlation of hemodynamic responses, right?
Right.
So this is the time scale that we're talking about. Exactly. Well, more than that, right? Yeah, more than that. More than a second.
All right, so I was actually saying.
Cara, tô levantando a mão aqui, ó. Será que não tem? Essa aqui não vale nada, não?
Ei, posso como mediador, posso falar uma coisa primeiro?
Não, tá, desculpa. Tá, tô esperando aqui. Só não deixa eu fazer uma perguntinha.
No, eu acho assim. In fact, like, let me go back to English just to close it. Wow, Braulio, what an amazing talk, and how such a nice way to close this event. Almost like I don't want to let anybody make any questions, because I want to finish with those last thoughts that you gave us. That's how, touched I was. I didn't cry like Siddhartha, but almost got there. Thank you. I really, really appreciate it.
Puta merda, esse é o único que ia fazer. I would be the only one that would be a neuroscience question here. You just guys, too spiritual here.
Right, however, if you want to ruin this moment, Richard, so please feel free to actually go ahead and do it.
No, I do. Sorry, because it's like, one thing that you guys don't notice, if Braulio reads the regulamento da Junta Comercial de Natal, it would be beautiful. And it's like, it's not because of, it's hard to know if his talk was good or if Braulio, gave his Braulio thing that makes the talk good, right? So Draud, I want to know one thing. It's like, okay, so you said that the visual cortex is kind of like, see their own thoughts. So let's say if you get an animal like, that doesn't rely much in eyes, for example, I don't know, like if you smell your own thoughts, like, I don't know how dogs dream or how mice dream, if it is. And I mean, I just like, my question is how much you think that this is, this fMRI thing you see, because it's just like a hemodynamic process. It could be just an interneuron that is like, increasing blood flow that has some specifics, like specific receptors or whatever in that region. How much you think that's actually you're seeing your own thoughts, or it's something that is from the visual cortex, it's something cellularly related to the visual cortex, something more reductionist? Yeah.
Well, it could be. I mean, like I told you, it's just a hypothesis that we are pursuing, and we don't have enough evidence to say that this is what's happening. And this is why I presented that as a hypothesis. But there is another piece of evidence, Richardson. But before I get to that piece of evidence, I just want to comment something on your question, which is, in the beginning of your question, you said humans. So humans are visual. You're right. And we have been discussing, and we talked about this. To me, it would be amazing to test this hypothesis in other species. So to me, it would be amazing to see whether or not this is the reason why the visual cortex is being modulated in the case of psychedelics in humans is because we are visual species. So does the same thing happens in the rat? This would be something that I'd be really interested to understand. Is it the somatosensory cortex that gets a boost instead of the visual cortex? Or is the olfactory system that gets a boost instead of the visual cortex? So this is something that we talked about, and I really think that this could be interesting. So answering your question, the piece of evidence that I want to mention is the decrease in the synchronization that you presented for 5-MeO-DMT. So a silencing of the brain in some sense. This is a data that we also have in humans, not only with, we have that on ayahuasca, on NN-DMT, on psilocybin. And curiously in humans, what you see is a significant decrease, particularly in alpha. There's a huge decrease in alpha. So basically an inhibition of alpha processing. The idea there and the speculation there is that there isn't also a speculation that the alpha rhythm in the visual cortex would act as an inhibition rhythm in order to prevent us from seeing things when we close our eyes. It would be very disturbing for sure if every time we close our eyes, we had a vision. So one of the perspectives, and there are published papers with that perspective, is that the alpha rhythm, when you close your eyes, you have a huge, in humans, when you close your eyes, you have a huge increase in alpha, almost immediate increase in alpha. So the idea there is that this increase in alpha, when you close your eyes, is there to inhibit your visual cortex. What happens during the experience with psychedelics is that this alpha that you see, the whole thing diminishes. So you don't have an increase in alpha when you close your eyes. Alpha, it's still there. I mean, you don't have an increase in alpha when you close your eyes. It's gonna stay as you saw in your own data, and we saw in our data. So this is the other evidence that I didn't have the time to present, Richardson, that might be another physiological link between seeing our own thoughts and the experience with psychedelics. But of course, it can be an artifact. I mean, we were talking, this is the biggest disadvantage on working with humans is, as compared to other species, is that our techniques like fMRI and scalp EEG, they're much less sensitive and specific than the techniques that you guys use.
Obrigado, Draud. Agora pode fazer todo mundo chorar. E, by the way, yeah, your beard is super nice, man. It's like, I'm jealous.
Perfect. I think Claudio was trying to ask a question earlier. I don't know if he still wants.
No, just to say that I see that I cried too. So
Yeah.
thanks for this nice ending. Thanks everybody. Thanks Kerstin. Thanks Tarcísio for organizing such a nice meeting. Although I was been sitting the whole day, it was great to be here with you remotely. And thanks everybody for making the Institute a better place to work, my best place of working, my best working place. So have a great weekend, everybody. So nice to be here with you. See you next time. Bye.
Thank you. Yeah,
Thank you, Tarcísio, for getting us through.
Again, me, Diego, Gabriel, and like a whole lot of people that worked on this. Diego worked super hard on building the replica. We had help from many people. Davi, Laisa, that built the artwork and so forth. And thank you to speakers, meaning, for instance, this is nothing without the people that actually present their work here. So this is probably the most important thing. And we went from some amazing talks from the students yesterday, were brief, but they're great, to this fantastic. We made Siddhartha cry twice or 2 or 3 times throughout this 24-hour period. So that's great. Not great. Not that I want to make you cry, Siddhartha. That's not what I'm saying. But I think we did some remembering, and like Braulio brought it up in such a beautiful way. We relived some very good memories that we were going to relish and carry with us for the next 10 years. And then we'll do it again, and we'll laugh, we'll cry, but we'll certainly enjoy the ride, the trip. Thank you. I don't think anybody should say anything after what Braulio said, so that's why I was trying to clap. But thank you all. I don't know if Kerstin wants to say a few words to finish it. Well, I don't know if Diego wants to say anything before we finish. And otherwise, we have a bring your own beer happy hour, so we can drink a beer together, either here or on Gather, whatever you guys want. Siddhartha has a class to teach, so he's not going to do that. But this is our next final event, a virtual happy hour. Right, Diego?
Yep. No, so thanks everybody for joining. Yeah, it's been great. I mean, I had a very good time. The other thing, I think our reservation actually expired, but it seems to be working. So whoever wants to just hang out over there, I'm gonna drop over there now. But I don't know. Final clap. I like the suggestion from Junior. Everybody wanna clap for everybody. Around.
Fantastic. Whoever's in the Institute, if you go to my lab, there are a few beers there. You're welcome to, grab a one and and have one. So thank you all. Let's move back to Gather. Thanks. And I'll see you on the dark side. Oh, actually on the Gather side.
Iguais, axé.
Axé.
Pessoal.
Até já.
Braulinho.
Valeu.
Pô, muito bom, hein?
Vamos lá, vamos tomar uma cervejinha lá. Eu vou
vamos. Se encontramos lá.
Se encontra lá.
Ah.